Okay, hey there. I'm just making this video to where I'm gonna just talk about this interesting paper that I think is pretty interesting and relevant to the RAS section of the course. And as far as like a discussion section for Friday, I've posted a copy of this paper to the Canvas website, and you can read it. You can ask me questions on Canvas. I made a plan to be on Zoom for some window of time on Friday, and so if you have questions about this paper, things you want to discuss, ideas that you got from it, I'll just kind of have like an open Zoom room and you can come and chat with me about it. But of course, also feel free to uh, send me emails or through Canvas if you want to discuss this paper a little bit. So what I thought I would just do is I would kind of walk you through what this paper is about and why I thought it was cool, and a few questions that it's left me thinking about, and maybe that'll seed some ideas for you for your own discussion points. Um, right. So, uh, as I said, this is all part of our week-long discussion about RAS signaling. And so, as I indicated, I have a plan on Friday to be available from 11:30 to 1 if you have any questions about the material or the paper. And as far as the paper goes, it's called Using Optogenetics to Interrogate the Dynamic Control of Signal Transmission by the RAS-ERK Model. And this is work that was done by Jared Tocher when he was a postdoc in Wendell Lim's lab. He's now a professor at Princeton who's continued to use this optogenetic approach that I'm going to discuss with you to do some really cool science. So, what's the idea behind this paper? Well, as we've really hammered home this week in the class, RAS-GTPase signaling is defined by the molecule RAS, which exists in an off state when it's bound to GDP and an on state when it's bound to GTP. And there are GEF molecules that can turn RAS on, that is, put it into the GTP bound state. And there are inactivators called GAPs that can convert RAS-GTP to RAS-GDP. And when RAS is in this on state, it interacts with downstream effectors like RAF kinase to drive signaling. And the kind of canonical pathway that we discussed and that I kind of mentioned that you should have a good grasp on is growth factor signaling, in which growth factors bind to a receptor and triggering that receptor tyrosine kinase to undergo phosphorylation of these tyrosine residues, which in turn recruits the GEF son of 7 list to the membrane via this adapter GRAB2. And once SOS is at the membrane, it can activate RAS. And once RAS is activated, it will recruit RAF to the plasma membrane, activate it, triggering this MAP kinase cascade that ultimately leads to phosphorylation of ERK and translocation of ERK from the cytoplasm into the nucleus, where it goes on to activate genes, so on and so forth. And I kind of chose this paper because it looks at RAS in the context of this very canonical system, but in a more interesting, sort of detailed way. And one of the interesting things that we discussed earlier in the week was that this paradigm of RAS to ERK signaling can actually produce different types of responses when different receptors are signaling, or when different receptors are activated. So, for example, we talked how NGF, when you stimulate cells with that, leads to a sustained activation of ERK signaling. Whereas if you stimulate through the EGF receptor, you get this transient ERK activation, which drives a different response proliferation compared to what you get with NGF. We talked about how this had to do with some negative feedback loop that downregulates receptor activity. And so right away, it seems like something interesting goes on in RAS signaling, where depending on the type of dynamic signal that is produced, at least at the level of activated ERK, you end up with different uh, cell biological responses. In one case, you differentiate into neurites, and in another case, you proliferate. And so this temporal component to RAS signaling seems to have a really important impact on cellular decision making. But, I just showed you two examples, NGF and EGF, and comparing the different dynamics produced by different receptor types can be complicated, and it can be hard to do in a very kind of rigorous and systematic way. And so, is there a way to systematically probe the effect of RAS signaling dynamics on cellular decision making so that we can reliably define the rules for how dynamics might affect signaling so that we can get a better understanding on this temporal component uh, to the cellular decision making. And so that is what the authors of this paper set out to do. So again, the paper is called Using Optogenetics to Interrogate the Dynamic Control of Signal Transmission by the Ras-ERK Module. And I sort of hinted at this paper in the previous lecture, where the idea here is what if we could take an easily controllable input, something like light, where it's easy to turn it on and off for as long as we want, with as much amplitude as we want, and convert that easily controllable input into something that drove RAS signaling activity. Well, if we had such a thing, where basically we could shine light in any duration, pattern, and amplitude on a cell and drive its signaling, then we would be able to look at some of these ideas like how different dynamics lead to different cell fates in a totally different way, where we could create different stimulus regimes for the cell, which are called optogenetic tools, and ask how they sculpt cell fate. And this is kind of cool because instead of being wed to the very particular transient or sustained outcomes that native receptors produce, we could produce any type of signal that we want and ask what happens. And so to do this, the authors exploited their understanding of how RAS is activated to build a synthetic light controlled RAS signaling pathway. And here it's like where if you really understand the logic of RAS signaling, then you can think of totally different ways to activate the pathway. And so remember that in the native pathway, the key way in which growth factors lead to signal activation is that tyrosine residues get phosphorylated on the receptor, and that recruits the son of 7-less GEF to the plasma membrane where it can activate RAS. And so really the key control point here is the recruitment of SOS from the cytoplasm to the plasma membrane. And so the authors of this paper said, well, what if we could do that in a way that doesn't involve receptor tyrosine kinase signaling at all, and instead was something we could control with light. And so what they did is they used what's called an optogenetic pair. So these are two proteins that in the presence of a particular wavelength of light, phi and pif will associate with one another. And in the absence of that light, or in the presence of an inactivating wavelength of light, those complexes will fall apart. And so if you shine a light on these proteins, they'll come together. And they said, why don't we put one of those proteins at the plasma membrane and fuse the other protein to that son of 7-less GEF that we talked about before. And in this way, whenever we shine light on the cell, these two proteins will associate. That will lead to the catalytic domain of SOS being recruited to the plasma membrane, and that will trigger activation of RAS and, by proxy, the RAF to ERK MAP kinase cascade module that we talked about before. Okay, so this is, you can write this on your blackboard and it seems like it should work. But of course, does it actually work in the cells? Well, after some effort by the authors to optimize the way in which they configured this, they were indeed able to make it so that light could drive the core activity of RAS signaling, that is, ERK translocation into the nucleus. And so to see this in action, what you can see is that when the authors shine 750 nanometer light, or sorry, excuse me, 650 nanometer light, which will drive the association of these components, then you see a translocation of ERK from the cytoplasm into the nucleus. And you can see this quantified here, that if you ask how much nuclear ERK you see, in once they shine the 650 nanometer light, you see a marked increase in the ERK translocation that can be reversed when they shine the light off, and then put back on when they shine the light back on. And so now the authors have something that, at least with respect to ERK translocation, appears to be able to artificially drive this pathway activation using light. Now they show through a number of nice other experiments that this optical stimulation method reproduces a number of key features of RAS signaling. So for example, if they shine the light on cells, they're able to drive proliferation using sustained light. So you see cells moving from G1 into G2. Using those PC12 cells we mentioned in the previous lecture, they can actually drive differentiation of these cells from a blob state to a neurite state using light. And importantly, many signaling receptors communicate not only to RAS but to other signaling pathways as well. And so there are growth receptors like PDGF, which will activate both RAS and, as you'll learn in other signaling lectures, PI3 kinase. And one of the cool things about this optical system is that you can actually specifically target RAS, but not the PI3 kinase branch with this wavelength of light. And so what you can see here is that when you stimulate with PDGF, you get ERK phosphorylation as indicated by this blot. But when you stimulate with PDGF, you also get activation of AKT, a marker of the PI3 kinase pathway. Whereas when you stimulate with this opto SOS construct, you get activation of ERK, but you don't see activation of AKT. And so not only do you have light-based activation, but you've actually kind of insulated your inquiry away from ERK signaling, or sorry, from any competing signaling that's going on from things like PI3 kinase, and you're focused exclusively now on this RAS-ERK module. So then the authors proceed throughout this paper to do a number of interesting things. I'm not going to talk about all of them in this little synopsis, but they use this module to look at interesting aspects of cell signaling. And so one of the things that they could do that was actually pretty cool was that they could do a dose response experiment to produce basically a dose response curve for individual cells in the population. And so what they do here is they would pick a single cell in their population and they would stimulate it with different durations of light inputs. That leads to different amounts of SOS recruitment to the membrane. And then they could ask how much ERK got translocated into the nucleus when you stimulate it in this specific way. And one of the cool things that they saw by doing this was that, of course, qualitatively, all the cells responded to the light activation. So the more membrane recruitment you got of the son of 7 Liskeff, the more nuclear ERK you saw. But there were quantitative differences in how these cells responded. And the authors were able to interpret the variability in the signaling responses that they saw to variations in the molecular composition of different cells. So cells that had, say, a little bit more of RAF kinase or a little bit less of some critical component ended up having slightly different input-output relationships. And so this isn't something that's really possible to do, a single cell dose response curve, when you use native signaling ligands like EGF or NGF, because you'd have to wash out the receptor, relocate the cell, and restim. And so here you have kind of a clean way of looking at the single cell dose response curve. So this is kind of a cool experiment and points to how different different configurations of cells can lead to different signaling outcomes. Another cool thing that the authors did was really explore the differences in transient versus sustained signaling using their system. And all they had to do this, which is pretty cool, was stimulate cells with either, say, a native input like PDGF or different durations of light input. So, for example, a sustained constant light input or a pulse of light input. And then they could take different time points and kind of look how the signaling response evolved using an assay that enabled them to look at a variety of different downstream components that get turned on in response to these things. And something cool that the authors found was that using this assay, they could identify different classes of responders within the population that interpreted these different signaling inputs to the cell in different ways. So, for example, they found a number of inputs that responded to PDGF, but didn't respond at all to their optical inputs. And as I mentioned before, because PDGF activates PI3 kinase signaling as well, these may represent targets that require both RAS and PI3 kinase signaling to be activated. Then they were able to identify a class of inputs that were able to track both transient and sustained inputs. So these are things that, if you stimulated in a sustained way, would turn on in a sustained way. And if you stimulated them with a short pulse of light, they would come on for the pulse and then come off. And then you could find a number of interesting target genes here that were activated by sustained but not transient light. So after being stimulated for an extended period of time, these genes start to get activated. But if you only stimulate with a transient pulse of light, these genes never get activated. And so these are genes who basically can interpret the temporal dynamics of the inputs that are applied to the cell and distinguish transient from sustained. And using this approach, as well as other interesting things that we can discuss, but I'm not going to summarize, the authors were able to develop sort of a coherent model for this dynamic signal encoding um, uh, by RAS signaling. And so, for example, they found that very short signals, so less than 4 minutes of input, were just never converted into signaling outputs by the cells. Short signals led to the activation of fast responders, but filtering out of slow responders, kind of like what we saw in the last slide. Whereas long signals, long duration signaling inputs, would both lead to the accumulation of fast responders as well as eventually things that come on later in the signaling pathway. So these slow responders here. And so, this paper kind of just laid a groundwork for being able to really interrogate dynamic signal encoding in a really rigorous and quantitative way for the first time. And I'll just close out my summary of this to maybe seed some thoughts for discussion. But I think this paper raises some really interesting questions and ideas for future things to do, some of which Jared's lab has gone on to do. And one key thing is that the authors primarily used only one cell line for these experiments. And the way in which signals are actually dynamically decoded by the cells, according to their own results, may have something to do with the composition of different components of the cell, or the sort of network context in which these signals are happening. And so one could imagine that you actually use this system to look at the dynamic decoding schemes in different cell types or cell backgrounds, and then perhaps you could identify why some cell types end up being more sensitive to disruptions in RAS signaling compared to others. Because remember, in the previous lecture, we talked about how almost 100% of pancreatic adenocarcinomas have a mutation in KRAS, but less than 1% of kidney cancers do. And so something is different about these different cell types that's causing them to be more or less susceptible to RAS signaling mutations. And it would be interesting to ask if there's something about how dynamic signals are decoded in these cells that leads to different outcomes. And related to this, the authors observe but don't identify the mechanism of this dynamic decoding. So we saw a lot of examples of things that respond to sustained but not transient signals, but we don't really know why. Why does that occur? And so it would be very interesting to figure out the mechanism by which dynamic signals are decoded by the cell, especially because you could imagine there might be some disease or cancer mutations which actually are manifest because of a change in how these dynamics are decoded. So, for example, if you had a signal, something that was supposed to ignore transient signals, but was mutated to perceive a transient signal and turn on, that might lead to aberrant signaling that has phenotypes that are deleterious to the cell. And so those were just some thoughts that I had from the paper, and so I'd be happy to discuss your thoughts, ideas, and questions about this paper through Canvas, over email, or on the Zoom call this Friday. And so I hope to hear from you at some point soon. And then, if not, I've enjoyed sharing my enthusiasm for these RAS signaling systems with you throughout this week. Care.